So I'm going to start off by mentioning that there are several types of cells in our body. There's our muscle cells, there's cells in our organs such as our liver, our heart, and now we're just going to focus on the cells in our brain called the neuron. So this is the general structure of a neuron. We have dendrite, cell body, axon, and axon terminal. The action potential occurs along the axon, and if we zoom in at the axon, we have an image sort of like this. So this is the axon membrane, this is outside of the cell, and this is inside of the cell. Generally, when the cell is at rest, the concentration of sodium is more on the outside as compared to the inside of the cell. On the opposite side, the potassium is more on the inside of the cell and less on the outside of the cell. To maintain this concentration gradient, the ions need to move against the diffusion gradient, and that's what action potential is. If we look at the general graph of the action potential, it looks something like this. So when the threshold of excitation is reached, the sodium diffuses in, and that means the sodium channels also open. Because we're going from a negative number to a positive number here, the process is called depolarization. After we reach about plus 40 volts, the potassium channel opens and the sodium channel closes. And then as potassium diffuses out of the cell, hyperpolarization occurs. That means we're going from a positive number to a negative number. And this after this, the potassium channel closes. And then after we reach sort of a stable state, the resting potential is reached. And that's about it.